Myself, Dr. JIBRAN presents to you Simply Pathology, and today we are back with a very important session. Today we are going to discuss the practical Osce or the Viva session, okay, in relation to the soft tissues. This is the part 2 lecture. Part 1 we have already discussed previously, wherein I have discussed 4 to 5 important exam slides in relation with the adipocytic group of tumors. Today we are going to discuss the peripheral nervous system group of tumors, and we have brought today 3 important exam slides. So as you know, this is basically an Osce session wherein we are going to see the individual cases and we are going to discuss the important exam questions which are asked in the Viva. So please pay a lot of attention. So without wasting any time, let us begin the first important case. Now 40-year male is there with swelling in the flexor aspect of the upper limb. So in the exams you are going to get nearly 5 minutes, okay, for slide identification, and they are adding 1 more minute for writing down the description. Okay, so let us look at the slides right now. So what do you see? We see a tumor mass over here which is both hypercellular as well as they are showing certain hypocellular areas. So the tumor is basically biphasic, as we can appreciate. If we look at the hypercellular area, if you are looking at the hypercellular area, this is the hypercellular area. In certain areas of this hypercellular areas, okay, we see that there is the nuclear palisading. The nucleus has gone at the periphery of this particular area and they are forming palisades, okay, like a fencing. You can appreciate over here also, whereas in between you are having empty areas, okay. So this area, as you can appreciate, this is completely empty. And what is this empty area composed of? They are mainly composed of your cell processes of these particular Schwann cells, okay. So they are composed of the cell processes of these Schwann cells, okay, and the cytoplasm as well, which is very ill defined over here, as we can appreciate, okay. So this is the basic hypercellular area as we can appreciate from this particular diagram. Then we see the hypocellular area, as we can appreciate over here, this particular hypocellular area. What you can see, first of all, there are these there are the same spindle shaped cells with buccal nucleus, but what we also see over here is the presence of thickened blood vessels, hyalinized thickened blood vessels, okay. And the matrix, if you see the background, the background basically is comprising of a myxoid matrix, okay. So it's a hypocellular having a myxoid matrix background, and along with that you are having hyalinized thickened blood vessels, okay. So this is the hypocellular area, as we can see, and the S100 protein IHC that we have seen is showing diffuse positivity, okay. They are showing diffuse S100 positivity. So what is your provisional diagnosis? Yes, please do write down in the comment section. Yes, what is your provisional diagnosis? Yes, fast. Yes, so the provisional diagnosis is that of a schwannoma. It is that of a schwannoma, okay. So the next important question the examiner is going to ask you, enumerate the different peripheral nerve sheath tumors. So you should be very fast and you should explain in this particular manner that peripheral nerve sheath tumors can be divided as a non-neoplastic and neoplastic. So in the non-neoplastic conditions you are having reactive conditions like traumatic neuroma. Among the neoplastic conditions you have benign and malignant. Under the benign conditions you have neurofibroma, schwannomas. You are also having perineurioma and granular cell tumor, whereas in the malignant group of tumors, most importantly, we are having malignant peripheral nerve sheath tumor. This is it, okay. So if you just answer like this, the examiner will know that you have a fair bit of an idea. But in the WHO 5th edition, if you see, they have already enumerated all the tumors and they have added certain new entities as well, like over here, okay. So these are all the group of tumors, but I have just told you the most important ones to remember, like schwannoma, neurofibroma, perineurioma, granular cell tumor. These are most important ones to remember. Along with that, you should remember this MPNST as well as malignant melanotic nerve sheath tumor. At least this much you should say in the exam. Coming to the next question. What are Antony A and B areas? So basically, I am just going to discuss and describe the basic histological features of schwannoma, okay, and I will ask you and I will explain you what are Antony A and what are Antony B areas. First of all, let me tell you that schwannoma is a group of a benign nerve sheath tumor. Schwannoma is a benign nerve sheath tumor which is mainly circumscribed, it is encapsulated, okay, and it is predominantly nearly 100% it is composed of Schwann cells. So the predominant population in schwannoma is a Schwann cell. This is the word that you will say. Then when you start describing, you will say it's a biphasic tumor comprising of hyper and hypocellular areas. In the hypercellular areas there are presence of what is known as verrucate body. So the examiner will ask you, what is the verrucate body? This that I have described you previously, this is actually called as a verrucate body. That means means where the Schwann cell nucleus, okay, it is basically palisading, it is basically lining up at the periphery like a fence, whereas the middle empty areas, as you can appreciate, these are the empty areas, right? These are the empty areas. These empty areas are basically pale eosinophilic, and they are comprising of the cell process and the cytoplasm of these particular Schwann cells, okay, of these particular Schwann cells. The individual Schwann cells, if you see, they are basically spindle shaped cells, but characteristically they are having a buckled nuclei or tapering nucleus like this, okay. So they have a buckled or a tapering nuclei, very important point. And this is how usually the nerve sheath tumors, the cells of the nerve sheath tumors, they look like, okay. So this is one very important point that you should note. And along with that, this is the Antony A area, that is the hypercellular area. Now, if you see outside this area, this is a hypocellular area. Now, this hypocellular area are also composed of the same Schwann cells, but the population of the Schwann cells are less. Along with that, the matrix, the background, if you see, it is myxoid in nature. And if you see the blood vessels, they are thick-walled, hyalinized blood vessels, as we can appreciate over here, okay, as we can appreciate over here. This is the Antony B area, okay. So, this is all about the basic histopathogenesis or histopathology of schwannoma, okay. Now, discuss the different variants of the tumor. So what are the different variants that we are having? Very fast, we are first having the ancient schwannoma. So what is the ancient schwannoma? So any schwannoma where the amount of this Antony B area, now remember, let me just remind you, Antony B area is basically degenerative in nature, okay. So any schwannoma where the amount of Antony B area becomes more and the amount of Antony A area is less, so that it becomes very difficult for us to understand whether this particular schwannoma, whether this particular tumor is a schwannoma or no. Okay, so this is the first point. Second point is the nucleus will show hyperchromacity. So the nucleus will become hyperchromic, and they will have basically clumped nuclear chromatin is there, okay, and slight atypia is there. So that is called as ancient schwannoma. Till date, even in the WHO 5th edition, there is no clear-cut criteria for the definition of ancient schwannoma, okay. Now, what is schwannomatosis? Now, before I go into the concept of schwannomatosis, remember one thing, that schwannoma, that is a familial variety, can occur as a part of neurofibromatosis neurofibromatosis type 2, okay. So the schwannomas are basically associated with NF2 in the familial setting, and most of these, or nearly 100% of these neurofibromatosis, they are type 2, they are associated with bilateral vestibular schwannoma. So nearly 100% of the familial variety of neurofibromatosis type 2, they are associated with bilateral vestibular schwannomas, okay. But remember, always remember that remember that multiple schwannomas, if you see all the time, okay, all the time, if you see the multiple schwannomas, that is not suggestive that it is 100% related to neurofibromatosis type 2, okay. So remember one thing, there are certain groups of schwannomas, that multiple schwannomas, they occur independent of neurofibromatosis type 2, and such group of multiple schwannomas which are occurring outside the setting of neurofibromatosis type 2, they are called as schwannomatosis. And classically, the mutation, it is not in the NF2 gene classically. Most importantly, the mutations are present in the SMAR CB1 gene, also called the INI gene, okay. So mutation is taking place over here. Along with this, they are also having inactivating mutations involving NF2, but this group of mutations are quite distinct, okay, and this is called as schwannomatosis. So remember one thing, there is neurofibromatosis type 1 associated with neurofibroma, neurofibromatosis type 2 associated with schwannoma, and there is schwannomatosis which is associated basically with the mutations of SMART CB1 and INI, okay. This is a distinct, genetically distinct group of multiple schwannomas. So remember one thing, whenever you see multiple schwannomas, not always it is associated with NF2. In some cases, they are quite distinct and they have this classical SMART CB1 INI mutation. But still, having said that, these group of tumors, schwannomatosis, they also have certain inactivating mutations involving NF2 gene, okay. I hope this is clear. Next important stuff is discuss the IHC profile of schwannoma. Most importantly, they are positive for S100 and SOX10, very important. They also show positivity for calretinine, which is in contrast to neurofibroma. Now remember, what is the risk of malignant change in comparison with NF? Now if anyone asks, is there a risk? Yes, there is a definitive risk, but the risk of malignant change means that from schwannoma it will become MPNST, malignant peripheral nerve sheath tumor. So in comparison to neurofibroma, the risk of malignant change in schwannoma is very less. Okay, this is very important point to understand. And if the examiner asks you, if a schwannoma develops into an MPNST, then what is the type of MPNST that it is developing into? It is developing most importantly into epithelioid variety of MPNST. It is developing into an epithelioid variety of MPNST. Okay, now previously remember, did I discuss the variants? No, I have not discussed the variants. So let me tell you about the variants of schwannoma. So schwannomas can have different kinds of variants. Most importantly, we have discussed about the ancient schwannoma, wherein I have already said that the ancient schwannomas basically means one thing, that they are having less amount of Antony A, more amount of Antony B, that is more amount of degenerative areas, and they show severe degeneration, so much so that it is difficult to diagnose it as a schwannoma. So IHC become necessary for diagnosis. Secondly, the nucleus is showing hyperchromasia along with clumping of the chromatin. This is basically diagnostic of ancient schwannoma. Apart from that, we are having cellular schwannoma. We have a plexiform variety of schwannoma, which is quite rare. We are also having epithelioid variety of schwannoma, and lastly but not the least, we are having your microcystic or reticular variety of schwannoma. Now one very important point that we have to keep in our mind is that previously there was a type of schwannoma called as melanotic schwannoma. Okay, but right now we are not using this term melanotic schwannoma. In fact, a new term has been coined, that is a malignant melanotic nerve sheath tumor. When you are using the term schwannoma basically is sounding benign, but the nature of melanotic schwannoma wasn't benign. It was quite aggressive in nature in comparison with the schwannomas. So that is why WHO has now coined a new term for the melanotic schwannoma, which is called as malignant melanotic nerve sheath tumor. Okay, and after that we are also going to discuss, okay, what is a malignant melanotic nerve sheath tumor. So let us try and understand about the malignant melanotic nerve sheath tumor. Very hot exam topic. Okay, so what is the definition? Malignant melanotic nerve sheath tumor, it is a rare peripheral nerve sheath tumor composed uniformly of Schwann cells showing melanocytic differentiation, usually arising in association with spinal or autonomic nerves. It is variably associated with Cadney's complex and frequently shows aggressive clinical behavior in comparison with schwannomas. Okay, now very important, PRKAR1A mutation and loss of PRKAR1A protein expression are seen in majority of the cases. So the diagnostic molecular pathology is loss of expression of PRKAR1A. Okay, and the essential and the desirable diagnostic criteria. So desirable, as we have seen, this molecular criteria, if happens, is desirable. Essential is frequent origin from paraspinal or visceral autonomic nerve, fascicular to sheet-like proliferation of heavily pigmented, relatively uniform plump spindle cells, and co-expression of S10, SOX10 and melanocytic markers. Okay, so there is co-expression of both the markers, S100, SOX10 as well as melanocytic markers are co-expressed over here. So given its different biological behavior, it should not be labeled as a melanotic schwannoma. That is why WHO has completely renamed it as a new entity, malignant melanotic nerve sheath tumor. Now, lastly, coming to the differential diagnosis of schwannoma. So, what are the differential diagnoses? The most important differential diagnosis is neurofibroma. So, neurofibroma, in contrast to schwannomas, they do not have a capsule. Again, they are having a pattern of background collagen, which gives an appearance of shredded carrot collagen. Okay, and they are composed of mast cells. So if you remember, I told you schwannomas are predominantly composed of only the Schwann cells, whereas neurofibromas, in addition to the presence of these Schwann cells, they are also having multiple other components, like they are having perineurial cells, fibroblast, and even mast cells are present. Okay, remember, in case of schwannomas, in the conventional variety, there was alternating hypo and hypercellular area, whereas in case of neurofibroma, it is hypocellular throughout. So you are having myxoid areas too, without any hypercellular areas. Now S100, though it is positive, SOX10, though it is positive, but it is not positive as much as it is positive in case of your schwannoma. So there is a weaker S100 and SOX10 positivity. Why? Because neurofibroma is not predominantly composed of Schwann cells. There are Schwann cells, but which are forming only 30 to 40% of the tumor. Okay, even calretinine is not strong and diffuse like your schwannoma. So there is a focal calretinine. Very importantly, neurofibroma, they show strong CD34 lattice-like pattern. It's a DNA fingerprint or lattice-like pattern which is important, which is basically formed by the fibroblast which are present in the neurofibroma. Okay, and it is indicative of the presence of this particular characteristic neurofibroma architecture. So stronger CD34 and factor number 13A positivity. Okay, so all these stains, they IHC markers, they help us to differentiate neurofibroma from schwannoma. Then the second differential diagnosis is leiomyoma and leiomyosarcoma. They may show palisading and extensive degenerative changes in the form of hyalinization, calcification, myxoid changes, and ancient nuclear atypia. So they might mimic like a schwannoma. But very importantly over here, in contrast to schwannoma, in schwannoma, if you see, the cytoplasm is not well defined. So you see like an empty space which is completely pink in color, right? But over here, in case of leiomyoma, leiomyosarcoma, okay, the cells are elongated with eosinophilic fibrillar cytoplasm, and the cell membranes are quite distinct. Second important thing, in leiomyoma, if you see, the spindle cells, they have more rounded and blunt ends, whereas in schwannoma or any nerve sheath tumor, usually they are having buckled end. Okay, they are having usually buckled end like this or tapering end. This is another very important. And if you do IHC, these group of tumors, leiomyoma, leiomyosarcoma, are positive for SMA and Desmin, and they are negative for S100. Okay, then we are having the very important differential diagnosis, malignant melanotic nerve sheath tumor. Now very important, formerly it was called as melanotic schwannoma. Now I told you why it is changed now. Most important thing that you should see is the site of origin. They are mostly paraspinal in origin, and the tumors are usually heavily pigmented. They are heavily pigmented. Okay, and as we know about the IHC, we have already discussed, there is a dual positivity of co-expression of S100 SOX10 along with melanocytic markers as well. And lastly, the last important differential diagnosis is malignant peripheral nerve sheath tumor. If you see, compared with schwannoma, MP and ST, they have a more infiltrative growth pattern. They are hypercellular, pleomorphic with a high mitotic activity with areas of geographical necrosis. Yes, and even S100, they are having Schwann cell. It's not like they are not having, but the S100 or the SOX10 positivity is very patchy in the conventional variety of MP and ST. So this is all about, this is how your Viva is going to go in the exam. Okay, so I hope you have you have taken a lot of information from this particular case number 1. Now we will discuss the second important case, that is the case number 2. There is a 18-year-old boy with a large deformity.